Students, the next step in the non-cyclic photophosphorylation is the absorption of light by the photosystem I. We saw that what happens to the photosystem II, that light energy falls on photosystem II. The two electrons are excited from chlorophyll A, P680. The electrons accepted by primary electron acceptor. The electron hole is filled by the splitting of water molecule. The electrons reach plastoquinone, cytochrome complex and plastocyanin. As this is high energy level, this is low energy level. So as electrons move from high to low energy level, the energy absorbed by the electrons is released and is used by thylakoid membrane to synthesize ATP. Now the process of photosystem I is entirely same to that of photosystem II. What happens that light energy falls on antenna pigment molecules. Antenna pigment molecules channelize all the energy towards the reaction center P700. Electron is excited and is accepted by primary electron acceptor. The hole is created in the photosystem I, and that hole is filled by the two electrons which were excited from the photosystem II. Now look here, the first step is light energy is absorbed by the chlorophyll A. Now this is photosystem I, so the maximum range is 780 nanometer. Then what happens that electron is excited to the high energy level. You may see here that electrons are excited to high energy level. Then excited electron is accepted by primary electron acceptor. The only difference between photosystem I and II is of the filling of the electron hole. In photosystem II, the electron hole was filled by the water, while in photosystem I, the electron hole is filled from the electrons which were excited from the photosystem II. Okay, this is the difference. And two electrons excite from photosystem II, and similarly two electrons excite from photosystem I. Similarly, water also provides two electrons. Now, we will look at the electron transport chain of photosystem I. So, electron transport chain of photosystem I has two electron carriers. First is ferredoxin, and this ferredoxin, as you can judge from its name, ferrum means iron. So, ferredoxin is an iron containing protein. We also studied one more iron containing protein, which was cytochrome complex. So, do remember cytochrome complex and ferredoxin both are the iron containing proteins in the non-cyclic photophosphorylation. The second electron carrier is NADP reductase. Any substance which has ase in the end, ASE, that means it's an enzyme. So NADP reductase is also an enzyme in nature, but do remember that it is flavoprotein coenzyme. More specifically, it is coenzyme in nature, and the type of coenzyme is flavoprotein. I'm only telling you for the general information, but you only have to remember that NADP reductase is enzyme in nature. As you can see, it's ferredoxin, we represent it by FD, and this is NADP reductase. What happens that the electron excited from the chlorophyll A, P700 of photosystem I are accepted by primary electron acceptor, and then they pass on to a series of electron carriers. The first electron acceptor in photosystem I is ferredoxin. Okay, the electrons are firstly accepted by ferredoxin, and the last electron acceptor or the final electron acceptor in the photosystem I is NADP reductase. So electrons from ferredoxin transfer to NADP reductase, and do remember these are high energy electrons. Okay, these are high energy electrons. These high energy electrons are transferred to NADP plus with the help of NADP reductase to make NADPH. The two hydrogen ions which were produced in water also move along the chain. Okay, so these two hydrogens, these two electrons and NADP plus, they all combine with the help of NADP reductase to make NADPH. Got it? Now, how this NADPH synthesis occur? So, see, we have three components right now in chloroplast. First is NADP plus, which is already present in plant. The second is two electrons which were excited from photosystem I. And third is two hydrogen ions which were from the water. They also travel onto the whole Z scheme. Okay, so these three components are available to us and we have to synthesize NADPH. Now look here. Firstly, what happens that NADP plus adds one electron. You see that NADP plus is deficient in electron. Positive charge means it is deficient in electron. When it accepts the electron, it becomes NADP because one positive charge is balanced by one electron, so it becomes NADP. So one electron has been used from here. Now what happens next? That this H positive, one H positive from here and the second electron from here combine to form hydrogen. It is one proton and one electron, so they combine to form hydrogen. Now this NADP and this hydrogen, they both combine to form NADPH. So you see NADP plus has been used, One electron has been used here and the other electron has been used here. Only one proton has been used here, so one proton is still remaining. So NADPH has been synthesized. Now what is the next process? That this NADPH and one proton which is remaining here, this NADPH, sorry, here is NADPH. This NADPH and this proton, one remaining proton, adds to form NADPH plus H positive. We don't write it as NADPH2 because it is not hydrogen atom, it is only one proton. So we write it as separately as H positive. Now, the energy which were possessed by this high energy electrons is now transferred to NADPH. The hydrogen from water is also transferred to NADPH. Now, this NADPH provides hydrogen to carbon dioxide to make glucose. And as hydrogen is added to carbon dioxide, addition of hydrogen is reduction. So, this NADPH possess reducing power. So, this is the synthesis of NADPH. Now, here is the animation where you will get the concept how is the light absorbed by the photosystem 1 and how is NADPH synthesized by photosystem 1. So here you can see the photon falls on the photosystem 1, all the energy is channelized towards reaction center, electron is excited and accepted by primary electron acceptor, then it moves to a series of electron carriers and NADP reductase combines electrons, hydrogen ions and NADP plus to make NADPH. Now students, we will look at the summary of the non-cyclic photophosphorylation. Do remember that one photon only excites one electron. But in the whole process, we studied 2 electrons because you saw in the last slide that 2 electrons were needed to reduce NADP plus into NADPH. Okay, so 2 electrons were needed, so we used 2 photons. 1 molecule of water was used and 1 molecule of water makes 1 atom of oxygen. Then other molecule of water splits, as I told you earlier that this non-cyclic photophosphorylation is a continuous process. Okay, it occurs continuously inside the cell. So immediately the next water molecule breaks, both of the oxygen atoms combine to form oxygen molecules which is released in the atmosphere. We use 2 electrons in the whole process, we use 2 hydrogen ions in the whole process. Oxygen was produced as a byproduct, ATP was produced by photosystem 2 and NADPH was produced by photosystem 1. Now look at this animation, photosystem 2 function. Okay, electron hole is filled by water, electron is transported to a electron transport chain of photosystem 1, photosystem 2, electrons excited from photosystem 1 and then NADPH is produced in the end. So you see here it is a continuous process. You can see that it is occurring continuously inside the cell. So this is the summary of the non-cyclic photophosphorylation. Students, now it is processing time. I have designed some questions from you and you will answer those questions. Okay. So the first question is, what is the first product which is formed in the non-cyclic photophosphorylation? Okay, revise it. What would be the first product? So majority might have answered it as oxygen, but that is wrong. I told you earlier that this oxygen is a byproduct. It is not the main product. Okay, so first product formed in non-cyclic photophosphorylation is ATP. Okay. How many iron containing electron carriers are present in non-cyclic photophosphorylation? I mentioned it in the lecture that there are 2 electron carriers in the non-cyclic photophosphorylation which are iron containing. First is cytochrome complex and second is pyridoxine. Now the third question is, which substances are protein in nature in non-cyclic photophosphorylation? So you see primary electron acceptor is a protein in nature. Then plastoquinone, it is isoprenoid, it's not protein. Then cytochrome complex is iron containing protein. Then plastocyanin is copper containing protein. If you move to the photosystem 1, then pyridoxine is also iron containing protein and NADP reductase is an enzyme, so it is also protein in nature. So except plastoquinone, all the substances involved in the non-cyclic photophosphorylation are protein in nature. Now which photosystem is involved in photolysis? It's so simple that only photosystem 2 is involved in the process of photolysis, and photolysis helps to fill the electron hole in the photosystem 2. Now what is the mechanism of ATP production called as? So this is the most important point. Do remember that production of ATP during non-cyclic electron flow is called as photophosphorylation, while the mechanism by which ATP is produced is called as chemiosmosis. So this is the difference between phosphorylation and chemiosmosis. Chemiosmosis is the mechanism by which ATP is produced, while that production of ATP is called as phosphorylation. So chemiosmosis will be discussed in the later videos. Also do remember the order of electron acceptor. First electron acceptor in photosystem 2 is plastoquinone. Second intermediate or second electron acceptor is cytochrome complex, and the final electron acceptor is plastocyanin. Now in photosystem 1, the first electron acceptor is ferredoxin and the final electron acceptor is NADP reductase. Also do remember one more point, that the most important event in the light reactions is the production of ATP. Okay, because ATP has stored chemical energy in itself, and ATP will provide chemical energy from ATP will be transferred to the glucose. So ATP production is the most important event. First product is ATP and the second product is NADPH, while oxygen is a byproduct. So do remember all the points. Also write the point on your copy, and you have one task to do, that practice the diagram of non-cyclic photophosphorylation which has been mentioned in your book at least 2 times. And if you have practiced the diagram, then do tell me in the comment section. I'll really appreciate you. So students, this was all from my side. If you have got the lecture, have a big smile on your face. And if you got the lecture, like the video, share it and subscribe it. Be happy, make others happy. See you in the next lecture. Allah Hafiz.